In this series, I want to talk about the general principles for the interpretation of immunogenicity. Basically, when we have studies, although you can use it for interpreting immunogenicity in a particular patient. And in this part one, I want to briefly review what the immune system response is and basically focus on anti-drug antibody response. Avoid using opinions. As regards any subject we propose to investigate, we must inquire not what other people have thought. I'm going to read that again. We must inquire not what other people have thought, or what we ourselves conjecture, but what we can clearly and manifestly perceive by intuition, or deduce with certainty. René Descartes, in the Rules for the Direction of the Mind, just in case your Latin is rusty, this is rule number 6, written between 1626 and 1628. What René Descartes is telling us is basically saying, let's try not to follow what people are saying just for the sake of it. Let's try to see the facts, analyze them, and deduce with certainty. Men have sought to make a world from their own conception and to draw from their own minds all the material which they employed. But if, instead of doing so, they had consulted experience and observation, they would have had the facts and not opinions to reason about. Basically, what Sir Francis Bacon in The Great Instauration, published in 1620, is saying goes pretty much along the lines of what we first reviewed with René Descartes. And it is not just by chance that I chose these two quotes from contemporary authors of the time of illustration in Europe. This is a time when people were basically waking up in terms of thinking and not just follow what you are told and what you are told to believe. Make sure that you can have the facts, that you can have the different experimentation and data so you can draw all of the conclusions with a level of certainty. This is a list of some of the most common mistakes that I encourage you to avoid when interpreting immunogenicity. Immunogenicity equals anti-drug antibody test results. I've seen this written many different ways, but basically the most typical statement is if there are no anti-drug antibodies, the drug is not immunogenic. The anti-drug antibody assay provides unequivocal positive or negative results. Anti-drug antibodies always interfere with PK. You can see it written as if the PK is not decreased, there are no anti-drug antibodies in the samples. In other words, let's not waste money and time in testing them because everything is okay with PK. If anti-drug antibodies interfere with PK, it is always in one direction. In other words, people say anti-drug antibodies can only decrease the PK. Anti-drug antibodies always interfere with function. Another way of looking at this statement that I've seen written is if there is no decrease in efficacy, then there are no anti-drug antibodies. Again, why waste time and money in testing them? If the anti-drug antibody titers are low, there are no neutralizing antibodies. And finally, baseline anti-drug antibodies are not important. Another way that I've seen this statement is exclude these subjects who have baseline anti-drug antibody positivity from the analysis because it's not truly an anti-drug antibody because they were there at baseline. So with the talk on interpretation of immunogenicity in the YouTube series, I hope that I can go through all of these different questions and explain why, in my opinion, these are mistakes and these are not trivial mistakes. These could actually have some sort of rationale behind it, but when we look at the data, when we understand the biology, we realize that we have to really avoid making these mistakes, look at the data and draw the conclusions in every single study we look at, or if you're looking at your patient on an individual basis, what is happening there. And don't get stuck with statements that could be mistaken in that particular setting. So immunogenicity is the ability of a substance, in this case is going to be a drug, and basically it's a therapeutic protein, to elicit an immune response. So what's happening is that the drug will actually be seen by antigen presenting cells. They could be any kind of antigen presenting cell, like a dendritic cell, which you see in the bottom, and it digests the protein and then it presents epitopes depicted here. I put one red, one green, and different epitopes are going to be presented. The B cell is capable of also identifying the whole antigen, in this case the whole drug that we're giving to the patient, and also process the epitopes. And ultimately the T cell, which can only see epitopes, will be presented these antigenic epitopes and will start producing cytokines and stimulating further cells outside the adaptive immune system. So these are target cells, and it can be producing responses on either Th1, Th2, or Th17 responses, also T regulatory responses. For further details on these Th phenotypes, I have another series of videos that you can also look at for what is the meaning of these Th phenotypes. But let's say, for the sake of this particular slide, that once the T cell is presented appropriately with the appropriate co-stimulatory factors or MHC engagement, a particular antigen from the drug, this T cell will be active. And it's not only going to be active producing a cellular immune response, which is what this arrow is saying. This is a T cell cellular immune response, but it can also produce some set of interleukins and cytokines that further stimulate that B cell into maturing, into producing both activated B cells and memory B cells, and eventually producing anti-drug antibody induction. And that's what we call humoral response. So right here on this simplified cartoon, I drew here how the immunogenicity to drugs can involve the T cell with a cellular adaptive response, but it can also bring the innate system with macrophages, neutrophils, and other antigen presenting cells. Outside the adaptive system, we can have innate system involvement, and also, of course, the B cell with anti-drug antibodies. So this is, in a nutshell, the sort of a cartoon that shows that if a patient, this statement I mentioned, that it's a mistake we should avoid. If a patient does not have anti-drug antibodies, the drug is not immunogenic. Well, that's not true. The drug could be immunogenic and could be triggering immune responses that do not include the B cell response or the humoral response. So what is the innate and the adaptive immune response to therapeutic drugs when they are acting as antigens? It's really the same as when you have any kind of other adaptive and immune response. The relationship of the innate and adaptive immunity is critical for us to deal with danger. And the differences between them are critical to make sure that we have a system that is working and helping us thriving in life. So in the innate immunity, which is here on the left, what we have is identical receptors. In this case, we are depicting here the toll-like receptor, and this is taken from Abbas, Lichman, and Pillai, Basic Immunology. I love this book, and I encourage all of you to read it and to read. These three authors are fabulous teachers. So coming back to the receptors, we see it's exactly the same receptor on exactly the same cell depicted here in green on the top left. But each one of these three examples is identifying a different antigen. So basically in the innate system, what we have is receptors that have less specificity, and they combined a lot of different pathogens. The identification made by the innate cells are basically two kinds of antigens that they can identify. The PAMPs, or the pathogen associated molecular patterns. So when we have something that microbes have, like, lipopolysaccharide or mannose, I mean, several different sugars that the human cells have less amount of those, or none. These human cells have been able to identify some pathogen-associated molecular patterns that are not typical of the human cells, and that's the way that they can capture that antigen-bearing cell and deal with it. So they're not very specific, but they are actually super useful because they can deal with many different pathogens at the same time. The other kind of antigen they identify is what's called DAMPs, the damage-associated molecular patterns. So, when we cut ourselves, when we have a third-degree burn, there are many internal cellular components that normally should not be in the bloodstream that now are exposed, especially, from the nucleus. So we shouldn't have DNA, our own DNA is not circulating in the blood or being presented to the cell surface. So when these particular receptors are seeing elements that are human, but they shouldn't be there, so they're the product of damaged cells, they also can react to clean it up. On the right-hand side, for adaptive immunity, what we see is now such degree of specialization that now we actually have one distinct antibody or T-cell receptor that will very specifically identify a particular epitope. So there's one different per different antigen. And this is, in the adaptive system, is a lot more focused and a lot more specific for that particular antigen. Both systems work in wonderful balance to help us deal with danger. As I mentioned, in terms of the receptors, you can have the three different receptors in the innate system. And by the way, I do have another series in the YouTube channel that explains the innate system in more detail. But this is basically explaining when we have, the toll-like receptors, we have a family. I do have another talk on toll-like receptors. Mannose receptors, as I mentioned, and the NOD-like receptors, which are internal in the cytosol. These are the typical receptors that we see. One same receptor deals with hundreds or maybe thousands of different recognition patterns. And on the other hand, what you see there, by the way, in the cell membrane is depicted right there in a pinkish color with the sort of dark green edges, and the cytosol is in light green. And what you see here now is, as I mentioned, either IgGs that have been either mutated, or the T-cell receptors that have had recombination of the gene segments that allow this very particular specificity that makes these cells, either the B cells or the T cells of the adaptive system, now be mature, specific for the antigen, in this case a drug, and also become memory cells for that particular antigen. So in a final rows of this particular table, what we have here is the innate system is not clonal. It's basically it's identified in many different cells. As you can imagine already, I mean, this is present not only on let's say, cells of the innate system, but it's present in tissues that deal with this exposure to pathogens and danger all the time. And of course, the skin is a big one. We're exposed to sunburns, to cuts, to millions of different pathogens, and we can deal with it. It's a great barrier. So respiratory system, GI system, they are there, and they're really non-clonal. Whereas in the adaptive system, we have either, selection of different clones. So it's oligoclonal. You have clones that learn how to identify these different specificity for antigens. In our case, we're talking the antigen is a drug, and it would be a few, but they proliferate, they expand, and they're there to act with a very sharp and dramatic response. All right, so basically these are the two systems: innate system, less specific, more more present in many different kinds of cells and systems, whereas the adaptive system is the one that will give us more memory, and it will produce a sharper and more pronounced response that is very specific for the antigen. In terms of the innate and adaptive immunity time sequence, what usually happens is the antigen, which in this particular talk is the drug, enters the system. The innate immunity will first see it on different kinds of levels. So if it's by the skin, or if it was a subcutaneous or intravenous, you can have the different cells that are engaged, and they start acting against this particular antigen. You can see they phagocytize it, or they produce complement activation, and they start killing the cells also by the NK cells. This is a very fast response. It takes hours, and it can just remain as an innate immunity response. However, in some cases, these innate cells will act as the antigen presenting cells that then trigger an adaptive immune response by either just stimulating T cells into effector T cells. You have several different kinds of T cell activation and T cell phenotypes. I have other YouTube channel videos that explain this in more detail of the immune system response and the TH phenotypes, and also how the immune response is triggered. Or you can have T cells and B cells. B cells usually require T cell for maturation and for antibody production. So again, here what we're seeing is that if we have an immunogenic response to a drug, it can have a response on the innate system only and never produce anti-drug antibodies, or it could have an innate immunity response that triggers only a cellular response with T cell activation, no anti-drug antibodies, or it can have the innate immunity that triggers the T cells that actually will communicate with the B cells into maturing them, and this is the one where we will see anti-drug antibodies. The timing is days after exposure. So it is very important when we are analyzing potential clinical manifestations itchiness, pruritus, hives, any kind of manifestation to the drug administration, we need to take in account this timeline. So having a subject having a reaction 4 days after an injection is still within the time frame of a potential reaction to the drug. So we should not discard it. It could still be an injection site reaction or an infusion related reaction if it's days after, because not all of these responses are going to be immediate responses. They could be delayed responses, especially when the adaptive immune system is involved. Talking about the B cell response to potential drugs. So what happens is when we have the drug, the antigen, the therapeutic drug, it can trigger a B cell response via the antigen presenting a particular epitope to the T cell, or directly. The B cell can identify the therapeutic protein on its whole, on its own. It can also not only present the different epitopes, but it can also be triggered by these epitopes if the T cell identified them, and then it triggers B cell response. So the B cell has the ability to identify the whole protein, the whole drug that we are administering, and also the epitopes that are presented by the T cell. The T cell communicates with the B cell. B cell will really not become active without T cell cytokines that stimulate further growth of this B cell. And the B cell will then create an activated B cell and a memory B cell. I also have other YouTube channel videos that explain the immune response, triggers, and sustainability in more detail. But going back to the drug, we're administering a drug that's a protein that has the ability to mount an immune response. And in the case of the B cell, it can come directly or it can be via the T cell. It does require T cell activation, and eventually this B cell response is going to be characterized by antidrug antibody induction. As I hope that you can see in there, the antibodies are binding the whole protein in many different places. So when we look at anti-drug antibodies, I want to first discuss a little bit or touch upon the structure and function of antibodies, because this topic is a bit complex, especially if our drug is an antibody and we have an anti-drug antibody. It's an antibody to an antibody, and we can get lost in our chain of thoughts because it's almost like looking into mirrors that are facing each other. So we just need to pause and identify what exactly are we seeing, which of the two antibodies are we talking about. It is important to know that when we have a therapeutic protein, a drug that is antigenic, we have a variety of potential structures that we're dealing with. So if you look at this particular cartoon, the first list there, aspirin is not a protein, of course. It's just starting from the size of the aspirin, considering that 1x size, which is 0.18 kilodalton, you can see the proportion of the size just in terms of the molecular weight of the potential drugs that we use that could be produced in bacteria or yeast on the left, and you go from the smallest, that could be a peptide like calcitonin, to proteins and eventually more complex proteins that have no sugars because they are produced in bacteria or yeast. The fact that I'm highlighting the element of sugar is because it adds a complexity to the quaternary structure of that drug, and it does produce potentially more antigenic elements on this particular complex protein. So the more complex it is, the more probability we have of having a particular part or epitope of that drug to be seen as a potential danger signal by the immune system and mount an immune response to it. When we move to the mammalian produced drugs, now we have the addition of potential glycoproteins, sugars added to this protein. And as I mentioned, that adds another level of complexity into the structure of these drugs that can potentially be seen as immunogenic. Now, don't interpret this as saying that the more we go to the left, the less immunogenic they can be. The immunogenic response could be infrequent, but it could be lethal. So the fact that you can have a rare instance of an immune response to a smaller molecule because there's less elements that can actually mount an immune response, it doesn't necessarily mean that that immune response in particular could be dramatic, could be a serious adverse event, and it could be death-threatening event. So the reason why I'm putting this here is just to explain there's a complexity to these drugs, but ultimately the response of the immune system is idiosyncratic, and we need to make sure we can interpret it as, in a case-by-case basis. In order to understand better the interactions between a drug that's a protein drug and antibody function, I just want to go through this cartoon and walk you through it in a logical way. So we normally have a cell with membrane receptors, and you have a situation with a soluble ligand that binds a membrane receptor. So when we use a drug to block this particular interaction, the drug can be either an antibody, depicted here as the green and blue molecule, or it can be a non-antibody protein, depicted here with orange and dark red. And that particular drug can bind either the soluble ligand or the membrane receptor. Now, the important thing that I'm showing in this particular cartoon is where they bind. So let's look at binding the triangular image there, which is the soluble ligand. What I'm putting in here is that the drug is binding precisely the piece of that soluble ligand that identifies and engages with the membrane receptor. So our drug is acting as a true blocking agent of this particular function. On the other hand, if our drug is now binding a membrane receptor, what I have depicted here is a binding exactly in the groove of that receptor that literally is occupying the receiving piece of the membrane receptor, and it doesn't allow the soluble ligand to work. So this is, in a nutshell, what happens with our drugs that are protein drugs. I just have to mention that the effect of that binding on the membrane receptor does block the soluble ligand from engaging it. But the resulting effect on the cell could be either an inhibition of that pathway or a promotion of that pathway. So depending if we actually have analogs to the endogenous proteins that will engage that receptor and make the cell more active, or we have inhibitors that bind that particular receptor on the membrane and could actually inhibit the final effect that the soluble ligand engagement normally produces. But for the sake of this particular explanation, the important thing that I'm showing here is that the drug blocks the binding of the soluble ligand, the natural soluble ligand, to the membrane receptor. So now we have a drug, which can be an antibody, or it can be a protein. And the antibody here is depicted in green and blue. the therapeutic protein in orange and dark red. So it is binding either the soluble ligand or it's binding the membrane receptor. But now we have antibodies to the drug. So when that antibody to the drug is binding the site that is the functional site of the drug, the site that precisely will produce the binding either to the soluble ligand or to the membrane receptor, what happens is that we do not have that blockade anymore. And then the soluble ligand is able and capable of binding a membrane receptor. Both of them are now free to work as usual. So an anti-drug antibody that is binding the functional part of the drug, either an antibody FAB portion or the piece of the protein that actually is binding the ligand or the membrane receptor, this anti-drug antibody will neutralize that particular function of the drug. So it's as if we were not giving the drug at all. The normal interaction will be happening without any effects. And this is the case when we have neutralizing antibodies. Now, antibodies have other functions, not only the binding of their target. And I want to review those functions because they are pertinent to both cases. When our drug is an antibody, we may have other functions of that antibody outside the antibody binding site, which is the FAB portion of the drug. But also, if we have anti-drug antibodies in both cases, these antibodies also have functions that are outside their binding site. So let's review first the complement-dependent cytotoxicity. When an antibody binds the surface of a cell and produces a cluster of receptors of that particular immunoglobulin, then this clustering will bring or will attract the complement complex, and it will begin the complement cascade of events that eventually literally drill a hole in a cell membrane and produce the death of that cell. So that is the complement-dependent cytotoxicity or cytolysis. So now I'm going to ask you to follow what I'm saying. I don't have animation for this particular narration, but let's now think that our drug is binding the receptor, but our drug is not an antibody. However, there's an anti-drug antibody to that drug. So the drug is bound to the receptor, and if it's not an antibody, it will not produce a complement cascade. However, if there is an anti-drug antibody to that particular drug that's bound to the cell membrane, we could potentially have a CDC effect that is derived from directly the immunogenicity of that particular drug. So, in other words, anti-drug antibodies could bind outside. They're not neutralizing because they are allowing the drug to bind the receptor. So the anti-drug antibody is binding outside the neutralizing site, but it is binding that particular drug that is engaged with the receptor, and it produces a cluster of receptors that triggers a CDC response, which is not to be expected. So that's the case when we do not have an antibody as a drug. Next, when we do have an antibody as a drug, it's a bit more tricky because it's as if part of the efficacy of that drug may be the CDC activation, like in the case of rituximab. So when you have an antibody to a receptor that will normally be not only binding the receptor, that's the antibody function, but through the Fc portion, it will actually activate the complement cascade and produce the cell death of the lymphoma cells, for instance. So this could be part of your mechanism of action of your drug. And if you have an anti-drug antibody that is not neutralizing, it is acting outside the FAB of your drug antibody. So your drug antibody is capable of binding the target, which is the receptor on the cells. But you have now an anti-drug antibody that could potentially be blocking the area of the Fc portion that activates the cascade of the complement activation. So in this case, your mechanism of action you would be expecting cell death, but by having anti-drug antibodies, you could have less efficacy, and they are not neutralizing antibodies. It's through the complement activation that they are decreasing the ability to have the mechanism of action working. So this is a very important concept, because when you do testing, we will see that in part 3 of the immunogenicity series, part 2, I'm sorry. When you look at testing for neutralizing antibodies, all we're testing is the ability to bind the target. We're not testing this ability. But this can cause either loss of efficacy and it's not neutralizing antibody, or it could also have effect on safety. The next function of the antibody that is through the Fc portion is what we call the ADCC. So again, I'm going to be doing the same kind of explanation as before. So we have a receptor on the cell, and let's say that your particular drug binds the receptor efficiently. And now you have antibodies to your drug. So if your drug is not an antibody, it would normally not produce antibody-dependent cell cytotoxicity, because this is dependent on the Fc receptor. And the Fc receptor in the effector cell, which is a killer cell that will release cytotoxins, and by those cytotoxins kill the cell, that Fc receptor is only present in antibodies. So when your drug is not an antibody and it is binding the cells, you would not expect that drug to have ADCC effects at all. When you have an anti-drug antibody and your drug is bound to the cell membrane, this anti-drug antibody could actually have an Fc receptor that engages with the effector cell and produces antibody-drug dependent cytotoxicity. So this can happen in cases where your drug is not an antibody. If your drug is an antibody, then this could be part of the drug's mechanism of action. Going back to the example of rituximab, this is one of the mechanism of actions of rituximab also. Not only the Fc portion can activate the complement complex, but it can also bind effector cells and produce release of cytotoxins and kill the cell by that other way. So when we have anti-drug antibodies, and the drug is an antibody that is supposed to have also ADCC as mechanism of action, the anti-drug antibody, which is not neutralizing because it's allowing your drug to bind the target on the membrane, when you have these anti-drug antibodies binding the Fc portion of your drug, they could block the ADCC effect that you would be expecting from your drug. So in this case, The effects on efficacy could be a decrease in efficacy, but it's not a neutralizing antibody. So to have a decrease in efficacy on particular drugs where you would be expecting them to produce CDC and ADCC cytolysis, your anti-drug antibodies could decrease that particular effect, and they could also be non-neutralizing antibodies. They don't need to be neutralizing to block this effect through the Fc portion. Similar situation happens with the other effect of the Fc portion, and this is in the case of an antibody that is binding a soluble ligand. So when you do have the binding of a soluble ligand by, let's go again with the same exercise, you have a drug that is not an antibody. It should not produce phagocytosis, but if you have an anti-drug antibody that's binding your drug that's bound to the ligand, you could actually have phagocytosis activation. This is going to produce certain signs and symptoms of immunogenicity, of course. So this is all immunogenic response to the drug, and these are all dependent on having a humoral response and anti-drug antibody in terms of B-cell humoral response. Now, the other thing on phagocytosis is again the same situation when your drug is an antibody and should be causing phagocytosis and phagocyte activation, and you have an anti-drug antibody that is not neutralizing, so your drug is bound to the ligand, but it is blocking that activation of phagocytes, then you have less phagocytosis and a decrease in the efficacy of this particular drug. So this is complex because we can have several different effects of antibodies, and when your drug is an antibody, you need to make sure that you dissect in your mind all the different steps that are happening so that you can understand how to interpret the data that you are seeing. There's basically 5 different classes of antibodies, and they are defined by their constant region chains on the heavy chain. So we have the IgG class, the IgE class, the IgD, IgM, IgA. There are features of each of them that are characteristics of these different classification. And you can actually read a lot more in detail about the different features and functions of these particular Fc portions. So now we are talking about the function of the Fc portion, which is the constant region of basically the heavy chain, and that's outside the antibody binding site. So going back to the interpretation of neutralizing antibodies or loss of function, they can actually be detached. You can have loss of function even if you don't have a neutralizing antibody. So when we spoke about the different functions of the Fc portion of the antibodies, it is important to identify that on the Fc region, there are 5 subclasses of IgG. There's the IgA, IgM, IgD, and IgE isotypes. And the ability to produce antibody-dependent cell cytotoxicity is more prominent with the IgG1, IgG3 subclass. The other isotypes will not actually have the ability to engage through the Fc receptors, the killer cells or the effector cells in the ADCC function. The complement-dependent cytotoxicity is going to be basically triggered with isotypic antibodies that are of the IgG3, IgM, IgG1, and to a lesser extent, IgG2 and IgA. So some of these isotypes will not be able to activate the complement cascade. And finally, for the antibody-dependent cell phagocytosis, what you see here is that basically all IgGs can potentially also induce phagocytosis as well as IgA antibodies, whereas other isotypes like IgM, D, and E will not have that effect. So this is important to know. Only when you have a drug that is an antibody, because that's the one you know for sure which is the isotype. So when you have a drug that's an antibody, you know the isotype and you know what the likelihood of that particular isotype having any one of these activities through the Fc portion is. You characterize that in vitro, so that you can follow if this is part of the mechanism of action or if this is something that you want to make sure you avoid. So then it depends on what is the kind of isotype that your drug is. And in all of the package inserts, when you are prescribing a drug for a patient or if you're doing research, you should know what isotype is the drug. And this is going to tell you some information about potential effects of that drug. The complication is when you have anti-drug antibodies, because they could block the effects of your antibody drugs if you are expecting these antibody drugs to have any of these Fc portion effector functions. But it also depends on the anti-drug antibody isotype. And that is idiosyncratic. We have no idea. It's most likely polyclonal or maybe oligoclonal response to the drug. So we could have a mix. Each individual is forming an antibodies to the drug, and we do not have control on what is that isotype. So that isotype could easily be one that engages equally well these functions as your drug is expected to do, and then basically, even though it's binding there, it may actually not have an effect on the expected efficacy. On the other hand, if the individual is mounting an immune response with anti-drug antibodies that are of a different isotype class from your drug, the effect that you would be expecting that particular isotype to have may be blocked. On the other hand, when your drug is not an antibody, we really cannot, we have anti-drug antibodies, we cannot really anticipate what the human isotype of that anti-drug antibody is going to be in all of the individuals treated. So we cannot expect to know if we have to test for one or another. But in part 3 of this series, I'm going to discuss how to generally look at the signs and symptoms that are important and why do we need to follow all of these special potential effects of an anti-drug antibody. Even when your drug is not an antibody, you can have, quote, antibody effects because of your anti-drug antibody. One function of the Fc portion of antibodies is actually binding receptors that we have in the liver. These are actually called the Fc neonatal receptors, and we all have them. Humans have them. I don't know if other animals do, now that I mention humans. Well, anyway, so humans have these receptor. And basically what's going to happen is on a first stage, these neonatal Fc receptors will identify the isotype on the IgG molecule. These are very specific IgG. They bind the neonatal receptor, and what happens is they go into the endosome together with other serum proteins. And as you can see there, the function of the endosome is to bring those other proteins to the lysosome for degradation. But what happens with everything that was bound to the Fc neonatal receptor, which were our IgGs, they are not going to be sent to the lysosome for degradation. They go on a third step into a recycling endosome and eventually go back into the blood. So in other words, there are some IgG isotypes and subclasses that bind stronger to the Fc neonatal receptor in our liver, and this increases the half-life of that particular antibody that we have in the blood. So the important thing about here is that if our drug is an antibody, we should know which IgG isotype it is, and we should have an idea of how this particular binding to the neonatal receptor is impacting and increasing the expected half-life of that drug. So we could have a drug that actually has been engineered to bind the Fc neonatal receptor with more affinity and be recycled more times. And in other words, we have a prolonged half-life of that particular drug. Or we can have drugs that bind with not so much length, but there are still different half-lives of the drugs, have a lot to do with this particular natural recycling that we have. If we have an antibody to the drug, and this particular antibody to the drug is an antibody to an IgG antibody, it could be blocking this binding to the neonatal receptor. And it could decrease the recycling and decrease the half-life. It could also be binding the IgG, but depending on the anti-drug antibodies, which we don't control. These are idiosyncratic, and each individual will mount an Ig response that is different. So if an individual is mounting a response with anti-drug antibodies that actually bind this neonatal receptor with more affinity, we could potentially be seeing an increase in the half-life of that particular, either the drug bound to the antibody, but definitely of this antibody in the drug, the anti-drug antibody. So it is very complex because this particular mechanism could be affected one way or another. Normally, if your drug is not an antibody, this particular metabolism doesn't play a role. But again, if you have anti-drug antibodies, they happen to be tightly bound to the drug when they get to the liver, and if they have the ability to bind the Fc neonatal receptor, they could be recycling the antibody and the drug into the system. So these anti-drug antibodies, either if they are to drugs that are antibodies or drugs that are not, can have more or less binding to the Fc neonatal receptors that can produce a perpetuation of an anti-drug antibody, not necessarily forever, it just prolongs that amount of time that you should be expecting to see the test positive in an individual for the anti-drug antibody. So it could be recycling that anti-drug antibody and increasing the half-life. Or they could potentially also be impacting how your drug, if your drug is an antibody, how your drug is normally metabolized, and it can actually increase or decrease the PK based on this function of Fc neonatal receptor recycling. So I want to summarize the uh, potential effects of anti-drug antibodies that we have reviewed in the talk of part one of interpretation of immunogenicity, and it's basically for clinical interpretation at the very end. But this part one was basically what are the elements that mount a B cell response and what should we be considering when we look at results. So because anti-drug antibodies are antibodies, they have an FAB, an antigen binding site, and an FC, which is a crystallizable fraction. If the therapeutic drug is endogenous and we have this antibody, the antibody via its FAB, its antigen binding site, can actually have long-term consequences if this anti-drug antibody not only binds your drug, but if your drug is homologous of an endogenous target. So if you have insulin and you have anti-drug antibodies, they could potentially be reacting with natural insulin from the patient. Same happens with any kind of endogenous drug. So this is something that is very important. I think I did mention in other parts of my talks that you could have a very low frequency for a particular anti-drug antibody to be present in a given drug, but the consequences could be tremendous. So this is what you need to understand in your particular system that you're working on. Is the drug that you're using endogenous? Even if the probability of having anti-drug antibodies are low, you need to make sure how are you going to follow up the patient, or how you're going to interpret the data if it's in a study, to make sure that if there are anti-drug antibodies, how do you make sure that you can characterize if they are reacting with endogenous target, and what are you going to do if that's the case, and what kind of safety surveillance you need to have for your patient or for the particular study. Now, if you have anti-drug antibodies to a protein that is not an antibody, the FC portion of the anti-drug antibodies can trigger phagocytosis, antibody-dependent cell cytotoxicity, and complement-dependent cytotoxicity. And this is especially if the endogenous target is a surface target and then your antibody is bound to it. So normally, if your drug is an endogenous target, is not an antibody, and binds the cell membrane, it should never produce any of these effects. Phagocytosis, ADCC, and CDC are only triggered by FC portions of an antibody. So normally your drug would not have any of these effects. In my talk, part 3 of immunogenicity interpretation, I will be explaining a bit about the different types of hypersensitivity associated with these effects that we need to follow up in the clinic or analyze in studies, either if our drug is an antibody or not. And finally, remember that also if the antibody to the drug has a particular FC isotype, it can bind the FC neonatal receptors, and it can produce a persistence of those anti-drug antibodies for a longer time than expected. If your therapeutic drug is an antibody, you need to do a bit more of thinking. I always think about mirrors that are looking at each other, facing each other. You need to angle one of them at least so that you can see better what is real, what's happening here. We have two antibodies. One is the drug, one is the anti-drug antibody. So what could happen? Via the antibody binding site, the anti-drug antibody can affect the monoclonal antibody FAB, so could be a neutralizing antibody. It could directly bind the functional site of the drug. But it can also bind the Fc part of your drug. And in that case, it can impact the ADCC, the CDC, and the phagocytosis effects of your drug. So if your drug binds, if your antibody binds a membrane receptor, and it's expected to produce antibody-dependent cell cytotoxicity, complement-dependent cytotoxicity, and or phagocytosis, depending on your drug's Ig isotype, if that is normally what you would expect as mechanism of action, an anti-drug antibody to the Fc portion could be decreasing the efficacy by reducing the ability of the drug to have these effects. Now, your particular drug may have been engineered to have less ability to mount these kind of Fc-dependent functions. And if your drug doesn't have this particular isotype, we do not control what the anti-drug antibody's isotype is. That is idiosyncratic, and it's each individual will mount different immune responses. So if a particular individual happens to mount an immune response with an isotype that has a high probability of engaging the antibody-dependent cell cytotoxicity, the complement-dependent cytotoxicity, and phagocytosis, any of these or all of these could now be seen in situations where you were not expecting this. So this would be similar to when your drug is not an antibody and you're not expecting to see these results. Bottom line is we do not know what the individual is going to mount in terms of the B cell response, what kind of isotype that's going to be. But on part 3 of the clinical interpretation series that I have, I will go over the clinical manifestations we do need to follow up and we need to analyze. I would assume that if needed, there are ways of testing these particular responses, but for individual case-by-case situations, it may not actually be necessary. It might be much more useful to look at the individual clinical picture. Via the FAB of the anti-drug antibody, they can actually bind the drug's Fc portion and affect the clearance via the Fc neonatal receptor. So this is another way that the anti-drug antibody can increase or decrease the half-life of the drug. And even if it's not decreasing or increasing the mechanism of action via either FAB or the Fc functions of cytotoxicity, it could increase or decrease the amount of time the drug is in the blood and ultimately affects efficacy one way or another, and also can have an impact on safety. Now, the anti-drug antibodies also have an Fc portion, and independently of where the FAB is binding the drug, this Fc portion of the anti-drug antibodies could also trigger those, phagocytosis, antibody-dependent cell cytotoxicity, and complement-dependent cytotoxicity if the endogenous target or the target of our drug is a surface target. So it's bound to the surface, and our drug may not be producing those effects, but the anti-drug antibody could be of an isotype that triggers that phagocytosis. So that's also an important thing to consider, that via the Fc, the anti-drug antibody can trigger those. And finally, also because the anti-drug antibody is also an antibody, via its Fc portion, it can be binding to the neonatal receptor in the liver and cause a persistent of those anti-drug antibodies for a much longer time than it should normally be. It prolongs the half-life of an antibody. To create this particular file and the animations and the cartoons, I have consulted many different literature reviews and different papers, but these are the ones that I would highly recommend, and I will put them in the YouTube description. Thank you for listening.